Hello class, let's continue to study chapter 3. We have studied the structure of gram-positive and gram-negative bacterial cell wall. In this lecture, we will discuss the mechanisms of gram staining and cell wall deficient bacteria. After Christian Gram established the gram staining in 1884, it soon became evident that most bacteria could be divided into two major groups based on their response to the gram staining procedure. Gram-positive bacteria stained purple, whereas gram-negative bacteria were pink or red. The difference between typical gram-positive bacteria and typical gram-negative bacteria is due to the physical nature of their cell walls. If the cell wall is removed, typical gram-positive bacteria stain gram-negative. Furthermore, bacteria that never make cell walls, such as mycoplasms, also stain gram-negative. During the gram staining procedure, bacteria are first stained with crystal violet, a dye with a positive charge that is attracted to the bacterial cell's net negative charge, and next treated with iodine. Iodine is a mordant that interacts with the crystal violet, forming an insoluble complex, and thus promoting dye retention. When bacteria are treated with ethanol in the decolorization step, the alcohol is sought to shrink the pores of the zinc peptidoglycan found in cells of typical gram-positive bacteria, causing the peptidoglycan to act as a permeability barrier that prevents loss of crystal violet. Thus, the dye-iodine complex is retained during the decolorization step, and the bacteria remain purple even after the addition of a second dye. In contrast, the peptidoglycan in typical gram-negative cell walls is very thin, not as highly cross-linked, and has larger pores. Alcohol treatment also may extract enough lipid from the outer membrane to increase the cell wall's porosity further. For these reasons, alcohol more readily removes the crystal violet iodide complex, decolorizing the cells. The counterstaining safranin, also a dye with a net negative charge, easily stains the decolorized cells so that they appear red or pink. After we learn about the mechanisms of gram staining, let's move on to cell wall deficient bacteria. The cell wall is the most basic structure of prokaryotes. However, there are cell wall deficient bacteria due to the long courses of evolution and spontaneous mutations in laboratory strains, such as L form of bacteria and mycoplasma. Besides, cell wall deficient bacteria could be reduced artificially by treatment with a chemical to disrupt the cell wall, such as protoplast and spheroplast. Some membranes of the genus Proteus, as well as other genera, can lose their cell walls and swell into irregularly shaped cells called L forms, named for the Lister Institute. When they were discovered, they may form spontaneously or develop in response to penicillin or lysergine. L forms can live and divide repeatedly or return to the walled state. Mycoplasma are bacteria that naturally lack a cell wall, although other bacteria require an intact cell wall to prevent the bursting of the cell. The cell membrane contains sterols that make it resistant to lysis. They tend to be polymorphic. They can be grown on artificial media. Mycoplasms are found in many habitats, including plants, soil, and animals. When a gram-positive cell is exposed to lysergine or penicillin, it will lose the cell wall completely and become a protoplast, a fragile cell bounded only by a membrane that is highly susceptible to lysis. Typically, a protoplast is spherical and is still capable of carrying on metabolism. When typical gram-negative bacteria are exposed to lysergine or penicillin, the peptidoglycan circulus is destroyed, but the outer membrane remains. These cells are called sulfuroplast. Okay, that's all for today's lesson.